Let me start the introduction of today's webinar. So hello, everyone. Welcome to today's webinar. I'm Selena, representing IBM Technology. I'm very delighted to have you all here today. So today we'll delve into the world of the cutting-edge imaging, focusing on the intravital live animal cellular imaging in immunology and transplantation. Let me introduce our guide for today, Professor Pilhan Kim, currently a professor at the Korea Advanced Institute of the Science and Technology, which is KAIST. So Professor Kim's expertise spans across the various research fields. He has an impressive background, including a research fellowship at the Harvard Medical School from 2005 to 2010. And in 2017, Professor Kim founded IBM Technology, a pioneering company dedicated to the innovative intravital microscopy and imaging techniques. A special thanks to our second presenter today, Professor Reza Abdi from the Harvard Medical School and the director of the Transplantation Research Center at Brigham and Women's Hospital. Professor Abdi's research focuses on the transplant resection and novel immunomodulatory strategies. He'll share practical insights into targeted drug delivery for the refractory disease, particularly in the field of immunology and transplantation research. Now, let's hear warm greetings from Professor Reza. So, hi, Reza. Could you share some warm messages with our attendees? Sorry, I think you're muted.
Thank you for inviting me. And it's been a great pleasure to collaborate with Dr. Kim. It's been extremely fruitful, and throughout my research, I always want to build something around live imaging, and having him around made things so easy. So I'm very grateful to Dr. Kim for his willingness and contribution. And I want to introduce this workshop, which is planned March 11th to 15th. And and through this workshop, you would get to know the plasticity, the scope of the work you could do with this machine, which is quite small, actually. One of the first things that came to my mind, so this is a very small machine, because I've seen live intravital microscopy, which takes probably the entire room. So so with that, I probably should start with Dr. Kim, presenting his data and scope of the work.
Okay,
Towards
great.
the end, there's a registration link. Selena asked me to mention it. In the end of it, there'll be a surveys for imaging workshop with registration link, so which you can proceed and said. Please go ahead, Dr. Kim.
Okay, yeah, thank you very much. Thank you very much, Reza. Thank you very much, Selena. So it's my great pleasure to have all of you here today. Okay, so my name is Pilhan Kim, and then today I'll talk about the real-time intravital microscopy, which is capable of in vivo live cell and drug imaging in a live animal model. Okay. Yeah, of course, I have a COI. So to declare, I'm a founder, CEO, and CTO of this company, IBM Technology. So we have this microscope installed in Dr. Abdi's lab in Brigham and Women's Hospital in Longwood campus. Okay, so I guess some of you already know what is intravital microscopy. So to give you just a simple idea, the intravital microscopy is quite useful and unique technique that can provide this kind of live video in cellular level in a live animal model in vivo. So in this video in the middle, this is the 10-minute video obtained from the lung of sepsis mouse model. So let me quickly just show you how we obtain this very high resolution, cellular level video from the lung of the live animal model. Okay, so in our group, so we implemented this small stabilized lung imaging window device. So in here, if you look at here, it has a small hole here, so we can suck out the air from the inside of this imaging chamber, so we can build up the negative air pressure inside around 930 to 960 millibar, which is enough to stabilize the surface of the lung against this transparent cover glass, as you can see in here. So now it's stabilized. Okay. After this stabilization, we still have some residual movement, which can be post-processed later. But anyhow, after this stabilization, we can obtain this higher resolution video from the lung of a live mouse model. So in this particular video, the green color is the blood flow visualized by intravenous injection of this FITC dextran right before the imaging. And then we also label the neutrophil, the intravascular neutrophil, by injecting antibody conjugate, anti-LI6 antibody conjugate with the fluoro, Alexa fluoro 6/7. So we inject those antibody conjugate at 1 hour before our imaging. And then those antibody conjugate very nicely label all the intravascular neutrophil inside the vessel, as you can see in this video. Okay. Okay, so I have many slides, so I just keep going. So, but if you have any specific question, please leave your question in the Q&A box in the below. And then later we can, I can answer your questions. Okay. But anyway, this is a 10-minute video, and then this, you can see very critical moment in this video, like at 7 minutes, right? So you can see this the intravascular neutrophil make a small cluster in here in the middle, and then it blocked the arteriole of this area of the lung in the sepsis mouse model, right? So we can, directly see the neutrophil plays a very critical role in disturbing the microcirculation in the lung of a sepsis mouse model. Okay. So, so I think, you already get some idea what you can do by using Intravital microscope. You can directly see how the cell behave and interact with each other in pathologic condition in the live animal model. Okay. Also, the microscope I use is capable of real-time imaging with the speed of 30 frames per second. So we can obtain 30 frames at 1 second with 512 by 512 pixels per frame. That is fast enough to see the flowing cells, such as breast cancer cell in this example. So this is a real-time video, as you can see. So actually, I slow down it twice, so it's half speed. But anyway, in this particular example, we label the vessel by injecting antibody conjugate again, anti-CD31 antibody conjugate to label the endocellular cell. And then we intravenously inject the 41 GFP breast cancer cell. And then we can see how this, the circulating breast cancer cell trapped in the capillary in the lung of the live mouse model, as you can see in here, right? So this is what you can expect by using real-time Intravital microscope to observe in the live mouse model. Okay, let me show you several more examples of what you can do by using a real-time Intravital microscope. So in this example, we are imaging the liver. Okay, so in this particular example, we use type 2 GFP mouse, which express GFP in vascular endocellular cell. So we don't need to label the endocellular cell in this example. So in green color, we can directly see the liver sinusoid endocellular cell. Okay, and then this is how we prepare our mouse model. So we make a small incision on the abdominal skin and peritoneum, expose the liver, and then put the cover glass on top of the liver surface, as you can see in here. And then after that, we catheterize the tail vein using some catheter tube. And then we intravenously inject the nanoparticle through the tail vein after process labeling. Okay. So when I started this real-time video, that's when we did the intravenous injection of the nanoparticles through this tail vein catheter. Okay, let me show you. Okay, this mouse is alive, so it's breathing. So when it breathes, you can see this changing of the this focus. But anyway, I hope you can see the flow of this presently labeled nanoparticle inside the liver sinusoid. So you can, directly see how this intravenously injected nanoparticle appeared in the liver in real time after the intravenous injection. And then you can also see how it's distributed, inside the capillary, liver sinusoid capillaries, as you can see in this video. So if you can label your drug with Prof, then you can directly see the delivery and distribution of that presently labeled drugs in the live animal model, like this example. Okay. So again, this is a real-time video, so we can directly see the delivery of this nanoparticle through the vein and its accumulation in the liver. Okay, let me move on. And then also, very recently, actually last year, at 2023, in collaboration with another professor at KAIST, Professor Young Yoon, we developed one AI method to remove the noise from the real-time video, as you can see in this example. So on the right-hand side, this is a raw video taken from the ear skin of this mouse model, H2B-GFP and Rosa 26MTMG mouse model. And then we also intravenously inject the red blood cell. So you can see this blue color is the flowing red blood cell inside the vessel. So in raw video, in real-time raw video, you can directly see this flowing red blood cell in blue color. And also you can nicely see the vessel and the nuclei inside of this ear skin. But by using this AI processing algorithm, we can very effectively remove all the noise from this real-time video. And this is the result. So we can nicely see how the tissue looks like in real time. So this is one example. And then it needs very heavy post-processing, but the outcome is very good, as you can see in this example. This work is already published, so if you're interested, you can find out the code in the website. Then this is another example, so you can directly see the result, the flowing red blood cell inside the vessel in the muscle. And this is another example. This is another real-time video obtained from the kidney, so we can nicely see the flowing IVC perfusion inside this tubular capillary in the kidney, and then we can distinguish the tubules, right? Very nicely, like this. Okay, so our microscope, the current model, it looks like this. So it has a single box design, but it's capable of both subconfocal and two-photon imaging in a live mouse model. And then it has a 4-channel two-photon detect, four-color, it's capable of four-color two-photon imaging by using integrated compact 920 nanometer pentasecond pulse laser module inside this box. Then it's also capable of confocal imaging, again, four-color confocal imaging by using this fiber-coupled 4-wavelength CW laser module. And then our microscope is capable of real-time imaging, as I explained before, with the speed of 30 frames per second at 512 by 512 pixels. Okay. And then the size of this box is quite small. It's about 85 centimeter in wide, and then 95 centimeter in this direction. So it's a small. The total footprint is smaller than 1 meter by 1 meter. Okay, so maybe I don't need to explain the working principle of confocal microscope, but just to show, the confocal microscope is capable of section the imaging of the 6 sample. Okay, so it's capable of very high resolution optical sectioning imaging of live tissue in a live mouse model in vivo. And this is just one example. Again, this is in vivo imaging data obtained from the inguinal adipose tissue of this adiponectin-free MTMG mouse model. So in here, in this tissue, so it's obtained from the, again, anesthetized mouse model in the adipose tissue. But you can see the, its resolution and sectioning capability very nicely. Okay, so you can easily obtain this subcellular level 3D tissue visualization in the live tissue, in the live mouse model. And then, in addition to that, in addition to confocal microscope, we can also use two-photon microscope at the same time. So in case of two-photon microscope, it uses longer wavelengths, near-infrared femtosecond pulse laser. So it's capable of deeper tissue imaging, deeper tissue imaging using fluorescence signal, right? In addition to that, also with the two-photon microscope, we can do another imaging mode, second harmonic generation imaging, which is particularly useful to visualize the collagen in the live tissue. So if you're interested, you can find a little bit more details in these publications, the review paper. But anyway, so we can nicely see the collagen in the live tissue, in the live mouse model by using second harmonic generation signal with the two-photon microscope. So it's quite useful to study the fibrosis mouse model. So this particular example was another in vivo imaging data obtained from the liver of liver fibrosis mouse model. In green color, it shows the fibrotic collagen detected by the second harmonic generation signal. Okay. Also, at the same time, we label the lipid droplet in the hepatocyte in the liver, so in red color, by intravenously injecting another Prof SF44. So this is just one example of how you can use the two-photon microscope to see the collagen and fluorescence signal in the live tissue in a live mouse model. Okay, and the 920 nanometer femtosecond pulse laser module integrated inside this box is quite useful because quite useful to do full color simultaneous imaging. So this 920 nanometer femtosecond pulse laser can excite both of GFP and some RFP. In this case, it's a TP tomato. So we image the, and then also we can detect the second harmonic generation signal simultaneously, as shown in this example. And then we can also see the blood flow by intravenously injecting other far red color dye. In this case, it's advanced blue actually. Okay. And then this data was obtained from the ear skin of the live mouse model, H2B GFP and Rosa 26 MTMG mouse. So we can see by looking at the green color, we can see the nuclei. And then by looking at the red signal, we can see the cell membrane of almost every cells. Okay. Then in white color, we can see the collagen. So if you are familiar with the microanatomy of the skin, this is the interface between the epidermis and dermis. So we can see many, cell nuclei in here. And if we go deeper, we can quickly, this is the dermal collagen in the dermis. And then you can also start to see the hair follicle in here. And if we go deeper, then we start to see the capillaries like this, and then more, and the hair follicle. And if we go deeper, like this, okay? And then if we go deeper, if you look at here, this is actually lymphatic vessel. Yeah, this is a valve, and then lymphatic vessel goes in this way. And then you can see this large vessel deep inside the ear skin. Okay. This is the four-color two-photon imaging data obtained from the skin. And this is another data obtained from the muscle. Again, same mouse model under anesthesia. So in this example, in the beginning, we start to see, okay, let me go back. So we start to see the fascia. Okay. This is the fascia, the collagen. And if we go deeper, we start, we start to see the sarcomere, myofilament inside the myocyte. So in here, you can see this periodic structure. That's actually sarcomere, myofilament inside the myocyte. And then also you can see the this big vessel and capillaries. This is muscle, and this is pancreas, okay? And this is kidney. Again, you can nicely see the tubules, structures, and capillaries, and spleen. Okay, all this imaging data was obtained in live mouse model under anesthesia. Okay, So in this for the mouse model, we can use our intravital microscope in almost every organ and tissue, as you can see in this slide. So we have been able to image the bone marrow, brain tumor, retina in the eye, skin, as I showed in previous slides, hair follicle, and various tumor model. This particular example is actually colorectal cancer model, pancreas, spleen, GI tract, adipose tissue. Also, we have been able to image the prostate and placenta in the pregnant mouse model, various lymph node, memory tissue, and then some cell transplant, kidney, muscle, liver, heart, lung, thyroid, and so on. So we have different, small devices and accessories that can be used to all these different organ and tissue in a live mouse model. Okay, let me quickly go through several examples what we have done by using our intravital microscope. So together with this intravital microscope, we can use several imaging chamber techniques, such as dorsal skin fold chamber. I guess some of you already heard about this technique. This is an imaging chamber that can be implanted on the dorsal skin of the live mouse model. As you can see in this video, this is our educational video. And in the middle, we can inject the cancer cell, normally human cancer cell, in the middle of this imaging chamber. And then we can keep this mouse to be alive for, for 3 to 4 weeks. And then we can observe how this, cancer cell interact inside the subcutaneous tissue. So this is one of our data. So again, our real-time intravital microscope is capable of directly see the flowing red blood cell in the tumor vessel, as you can see in this example. Okay, so in here, again, we inject anti-CD31 antibody conjugate to label the endocellular cell, so we can see the vessel. And then also we injected some red blood cell, fluorescently labeled the red blood cell with the ID. And then in blue color, actually, this is pseudo color of LSGFP cells implanted in the middle of this dorsal skin fold chamber. And then let me show you some published work. This is published in Small in 2018. So in here, in collaboration with one of the professors in Purdue University, they made this nanoparticle which can target selective in the activated tumor vessel endothelium. Okay, so they made this quinic acid derivative nanoparticle, in short QAMP, so it can target this selectin inside the tumor. Okay, and then this is how it looks like. And then what it did was we inject the QAMP nanoparticle after fluorescence labeling with Alexa-Flora 555 to this dorsal skin fold chamber tumor model, cancer model made by this triple negative breast cancer cell, MDAMB231 GFP. Okay, and then at the same time also we labeled the vessel by using anti-CD31 antibody. You can see here. So this is 2-hour, 6-hour, and 24-hour after the intravenous injection of this nanoparticle. Okay, and then you can nicely see how this nanoparticle delivery happen over these 24 hours. So at 6 hours, as we expected, we can see the increase of this nanoparticle, but mostly inside the vessel, so attached to the endothelium. Then at 24 hours, we can see the many this nanoparticle get out from the vessel and then delivered to the cancer cell, the single cancer cell, like this here, here, and then here and here as well. Okay, so again, so this is a very nice example. We can utilize intravital microscope to directly see the drug delivery in the live mouse model. But in this case, for 24 hours. Okay, but we can extend it even more. So this is an example, another example, the observation at day 7, 10, 13, and then we can find the exact same vessel with some treatment or without treatment, and then we can directly see the efficacy of this treatment. Okay, so again, this is one example, another example. So with treatment and without treatment, and then we image exactly same location at day 7 and day 13, and compare how their vessel changes over time with and without treatment. So this is a good example. We can evaluate the efficacy of the drug in a live mouse model for several weeks. Then it's another imaging window technique called abdominal imaging window. So it has a ring shape. So we made our own version of this abdominal imaging window, and then we can use it to visualize the abdominal organs such as liver, kidney, pancreas, spleen, and so on. Okay. So let me just share some of our ongoing work. So in this particular example, we try to image the pancreas for 2 to 3 weeks. So we injected this PANCO2 GFP cell into the pancreas in here after implanting this pancreas imaging window. So this is the same mouse imaged at day 1 to day 15. And then by using this abdominal imaging pancreas imaging window, we can find out the exact same location inside the pancreas. Okay, so this is day 9 and day 14. Actually, this is the same location, so you can see how the tumor vessel changes, and then how this GFP expressing pancreatic cell proliferate, grows. Actually, we can do it every day because we implant this in pancreas imaging window. So from day 9, 10, 11, 12, 13, and 14. So again, this is the exact same location, and then you can directly see how this tumor vessel changes. It very dynamically changes, almost every day. So if you compare here to here, you can see huge changes in here and also here. And then later we can see the huge change in this area. So we can see the vascular liquefaction here, and the over dilation, and then the vessel became very spiky shapes like this. Okay, so this is just one example. We can do repeated longitudinal observation in the disease mouse model in cellular level. And then we are trying to see label additional things like MECA79. So we inject the MECA79 antibody to find out the vessel that to find out the endocellular cell that express the ligand for these MECA79 antibodies at near these pancreatic cancer cells like this. And then, as you may know, so as I explained before, by detecting second-normalization signal, we can also see the increase of the collagen in this pancreatic cancer model, like this, from day 8 to day 16. So you can see this gradual increase of pancreatic cancer cell and then collagen depositions. Okay, so due to time limitation, let me quickly finish my talk here. Okay, so if you are interested, please visit our webpage, www.ivimtech.com. So in here, you can find out the specification of our microscope. Like so, we have a multiple model, comfort core microscope, 2-photon microscope, or dual mode microscope. And then we also have a customization options in case if you need handle the larger animal like a parrot. Then you can customize our microscope to handle those large animal model. And then we also have a lot of information, what kind of disease model you can use, and then what kind of quantification you can do by using Intravital microscope. And then we have uploaded many webinars on our web page with specific topics, like peptide nanoparticle imaging, or exosome imaging, or dry tract imaging, and so on. So if you have some very specific topic of interest, then you may be able to find the webinar, uploaded webinar in our web page. And then we also do some contract-based research services as well. So we have our in-house animal facility and in-house animal imaging lab. So we can do contract-based research service. So we have done it for mainly for the biotechs. These are the two examples. So we have been able to image the exosome delivery or antibody drug delivery in the brain or in the liver, respectively. Okay, so IBIN technology, we are providing this intravital microscope. And then we also provide contract-based research service. And then we have our own specialist team with a very long experience in intravital imaging, more than 10 years. So we can provide a very in-depth, scientific support to provide the consulting and then technical support for your, experimental design of intravital imaging. And then we also provide, various training service and technical service as well. Okay, thank you very much. So let me finish my talk here. And then later, I will answer all of your questions.
Thank you, Professor Kim. For the next, we're gonna hands over to the Professor Reza for the next presentation. Sorry, I think Professor Reza is muted.
Go to the next one. Do you have the control or you have the control? I have the control, right?
Yeah, you have the control.
I think, hold on a second. So I should be able to move it, right?
Yes.
I think Selena needs to give me the control.
Okay. Actually, it is.
Can you move to the next slide for me, Selena, maybe?
Yeah. It is controlled well right now. Yeah.
Okay.
It's great.
Good. Thank you. So I got to know Pilhan several years ago through a colleague of mine, and one of the biggest problem that we have in both obviously research and clinical field is that we end up giving immunosuppressive drugs that typically lead to more complications, and with that we started thinking, can we deliver some of these drugs to specific sites to be able to reduce the amount of drugs versus an increased efficacy part of it. So one of the main lines of research we are pursuing in our lab is to deliver nanoparticles. And one of the biggest problem with nanoparticle delivery is actually looking at the homing of it, which I was very interested to collaborate with Pilhan on that end. It's extremely important to be able to monitor these particles live through live imaging, because most of the static counting, etc., doesn't really recapitulate the actual delivery part. And I think this becomes a very important target drug delivery. And I was very much interested also when listening to Pilhan. Actually, I wrote so many questions, and we want to use this machine for more applications, because you mentioned that you can actually look into ADC, antibody drug delivery, and that's one big question I have in my lab as well, because can we see these antibodies homing, etc. So that would be a major milestone if you're able to do this. So I actually wrote it down, Pilhan, that I want to do those peptide, MP, and antibody delivery as well, which would be a great help for us. And most of you guys know about these particles, and there are various forms of particles, but I think one of the nice thing about imaging is that you can put different dyes to these particles and monitor the trafficking, and we make different forms of particles, but mostly we stick to the particles that are more translational to large animals, and staying away from those more cutting edge, more harder to translate type sort of particles. And a number of benefits you can get from nanoparticle delivery. And I think what Pilhan is trying to say is this one, that you get more target delivery basically with your antibody and with your particles. So we did some imaging of, we are interested in lymph nodes. So, but the images that he showed in lung and liver, those are really interesting ones that I didn't know that actually you can image lung that way. Is every time I listen to your webinar, I learn something new actually. Is quite remarkable how much you can. And I think the motion part that you have the AI part introduced, that's actually a very smart way to get these images more interpretable. And I think if you can access people to your AI person, who build a collaboration would be very helpful on the academic end. I think it would be greatly it would be instrumental. And we are interested in lymph node because lymph node is primary site of alloreactivity. And we put organ transplant in mice, and we interested in imaging heart and lung. And I assume if the way you image the lung, you should be able to image the heart transplant as well. And most of the delivery thus far for lymph node has been, going through lymphatics through skins. And we pretty much targeted a vein that exclusively expressed or present in the lymph node, and that's called high endothelial venules. And this is the part that we are interested to deliver drugs. And so I think this is I'm not sure what this video, but this is the image that we made with your microscope, Pilhan. So on the electron microscopy is classical T cell trafficking across this vein. This been reported in earliest report was 1950. As you know, Sergem Gound reported that T cells pass through this vein on a daily basis. And then on the left side is the image that we made with your microscope, basically showing beautifully the structure of high endothelial venules, and the resolution is very interesting to us. So we are adding T cells to it, and we are adding more other variables like dendritic cells. So we've been able to image some CD11, CGF3. So you can actually add more to it, right? Because one of the things that we discovered, which was very interesting, is you can actually introduce different antibodies. You can, if you don't have multicolor reporter mice, that, if you're interested in dendritic cells, T cells, in addition to stromal cells, you don't need to have all the reporter in one mouse. You can focus on one. If you're focusing on dendritic cells, you can get the GFP, C11C, or whatever there, and then introduce an antibody like collagen or antibody for vessel. So now you have multicolor images of the tissue. So basically, this is the structure of the lymph node, and these are the veins that we are interested in. This is the image of the endothelial vein that we are interested. So one of the things that we try to do is, this is a work that Samuk done in my lab. He was interested in trafficking of these particles through vein, and he came up with a gold nanoparticle, and shows that the gold nanoparticle passes through. But I was very much excited to see your nanoparticle live imaging, right? And I don't know whether you're able to look at the subcellular level. Probably not, because the resolution is not that high. But one of the question I always have is that when you get the nanoparticle, and you do targeted, you put antibody, etc., and I don't know anyone has been able to show live image that this particle, when it sticks to the endothelial cells, right, and because that's the first layer they need to pass through, then they go inside the particle, inside the endothelial cells, then they pass through. So one of the key questions would be to answer, which I think only way to answer is live image, or actually one of the methods is to see how these particles pass across endothelial cells. I mean, if you cannot do even, obviously look into subcellular level, they do pass through tight junction. In the liver, for instance, the sinusoidal cells are pretty much open. So it would be very cool to show that these particles actually can pass through those fenestration or not. So this is one of the struggle we've been, or one of the big question we have, which I'm going to actually pursue more with your imaging technique, is to be able to focus on endothelial cells and see how these particles traffic across between the endothelial cells or amongst the endothelial cells. That's the big question that I'm very much interested to use your imaging technique to answer. And I'm just going to go. So some of these imagings that we did, this is the bioluminescence imaging that we did for labeled nanoparticle, and we were able to show that they home to specific sites like lymph node. But I think these are very crude way of looking at it, right? So if I can put more live imaging to this particular lymph node and say, wow, you know, okay, particles are accumulating here, but how they're behaving, and it would be even fast, because one of the image that got my attention was that you said the particles were taken by cancers. So that would be extremely important, because if you can live image and over the period of several hours or days in anesthetize these mice and say, well, these particles pass through these vessels, because one of the biggest problem with nanoparticle delivery is that the initial idea was that these things, the particles pass through highly fenestrated permeable vessels of the tumor. But now we know that these vessels are not that permeable, right? Because there's so much fibrosis around it. So, and then because you can see the collagen in these tumors, so I was thinking that some of the big questions that we can answer is actually through live imaging, that how these particles pass through this collagen and how they get to tumor cells. So these are this was very interesting that I was, and we, you know, we can do a standard homing of these particles, like how over time. But again, I think what excites me the most is how to see how these particles being taken up by different cells, like not just by tumor cells, by antigen presenting cells. And the other question I had, maybe Pilhan, you can also, one question was, maybe you can explain after or during QA, is that how we can see antibody ADCs, how we can see small molecules that are very small. And can we also put an imaging technique? For instance, you put a chemotherapy drug. Can we show that live image that cells are dying? Like, you know, those are interesting things to do, because especially if you have a marker for apoptosis, for instance, right? Those are things that I was going to ask you. So if you have time toward the end, we can. So this is some of the stuff that we do with basically conjugating, and these are the images that we got with our nanoparticle, where we put an antibody inside the particle, and we try to image this the payload, actually, right? So one thing is to label the particle, the other thing is to label the payload. And again, this is a therapeutic. I think here we can see that they're picked by dendritic cells. But again, if you do a C11C GFP reporter, you can use different colors and actually see these things are being picked by different antibodies. So which would be very cool, actually. We show some survival benefits. I'm not going to go too much into, but we also did this delivery platform for in large animals. So you mentioned that the apparatus can be changed for larger animal, but this is not the size for like a larger than rat, right? I think right. Okay, so this is another question we had. I mean, the other question is that if we can keep some of these lymph nodes or tissue in the, you know, some people keep them on high oxygen with cultures, etc. Maybe it's a bit contrived, but that would be also a cool way to look at the particle homing in those given tissues from, let's say, primates. Even if you're interested in lymph nodes of primates, and whether taking them out and keep them maybe for a few hours and see how these cells pick them up, those would be very interesting. So we are doing a lot of target delivery to different. So the other thing that I'm very much interested in the pancreas, because we do drug delivery to pancreas, but I was quite fascinated that you can actually image the pancreas. Is that would be a major breakthrough for type 1 diabetes and drug delivery to those to that model. So this is another imaging that we did of pancreas, where we are looking for particles in the tissue. But again, and I recall you mentioned you had an image with islet as well. So, you know, if you're interested in type 1 diabetes, you're more interested in the islet area, and probably you need to put an insulin to it. I don't know how you would identify those live, but if you can image those islets live with the T-cell drugs, that would be really interesting to us, that we can image this pancreas of this type 1 diabetic mouse over time. So this is the image that we did with the MRI to figure out whether particles home to pancreas, but I think this was so complicated and so labor intensive to do MRI type things. But again, this is way more costly. I mean, the cost of putting the MRI machine, etc., was way more. So I think it would be more attractive to do a more live imaging the way you do it, and especially if you can image the pancreas over time. This is the image that we got from particle homing to the islet. And I think these are the images of payload again. Here we had antibody labeled. Here on the left side is the bioluminescence, on the right side is the imaging, but I think I'm probably not gonna go too much in the T-cell trafficking part. And so this is one of the images that Pilhan, we're very much interested. I'd like to develop is to image heart live, right? So we are we do transplants. So the heart is tricky, right, because of the pumping and motion. So even people who are interested in cardiology, they might be interested in various forms of cardiac models where your imaging would be crucial. So one of the things that I'd like to develop is to be able to image heart and kidney transplant. I saw your kidney imaging, which was very cool. So kidney probably is easier. We do kidney in mice. So these two organs, I'd like to develop imaging around them with your technology. And I think this is the nanoparticle that we deliver with labeled nanoparticle, and we can bioluminescence, and we can see. But again, these are very static type images, which doesn't really tell us exactly what is the and here one way. Again, I would love to know how you can image a drug, because we do drug delivery. So in the particle, you could put labeled drugs, whether we could image or not. So you can comment on that. That would be really important to us, because we do drug delivery to the organ prior to transplant, and to be able to suppress the inflammation early on post-transplant. So because post-transplant, what you do is you give everything intravenously, and it goes everywhere. So if you can localize the anti-inflammatory drugs in the organ prior to transplant, is extremely crucial to more effectively suppress inflammation. And I think I'm gonna go. So we did some long. This is the image. This is the intro long Pilhan injection of nanoparticles. So we don't need to image the primate, but I think we could pretty much image the lung in a preservation, because you can keep these tissues for at least 6-7 hours. Some of them are on pump for, let's say, 48 hours, right? So it would be really, really exciting if we can image the organ on a pump. I mean, the organ is going to be, or even organ on a preservation fluid for 6 hours, and you basically inject the particles and take the lung out and image them live, and look at the uptake of these particles by different cell types in the organ. That would be really interesting. So some of those things we are doing with lung. And I think we are doing antibody drug delivery. I'm going to stop here. So this is the part that I'm going to stop and have you comment on antibody part and small molecule part. And I'd love to watch your webinar on peptide delivery. That's very interesting. So probably I'm going to stop here and have Selena take over QA, and we go from there.
Okay. Selena, can you allow me to share my screen? Okay, so we have many questions from in the Q&A box, but so before answering those questions, let me just show these slides. So this is actually how the antibody drug looks like when we do imaging. So antibody is very small, so we cannot distinguish the individual molecule, but still we can see it looks like a diffused signal inside the tissue. If you can see here, so this is actually the antibody drugs. The upper panel is normal, just a naive drug, antibody drugs. And the lower panel is some modified antibody drug that can penetrate the BBB. So if you compare here at 24 hours and 48 hours, we can see more diffusion of this antibody across the BBB to the brain tissues. Okay, but anyway, this is how the ADC, antibody drugs like ADC will look like in the tissue.
Would you be, Pilhan, sorry to interrupt you, would you be able to zoom out on that structure and see whether how much passes through, or like, let's say, can you image, because this antibody should probably gets internalized and some of it probably leaked out, right?
Yes, yes.
You should be able to see those things too, right?
Yes, yeah. Maybe, yeah. If we zoomed in enough, and then if we select the light time point, then we should be able to see those processes.
Okay, got you.
Okay, so so now let me answer some of the question in the Q&A box. So I already answered the 2 questions, but the first question was, how long can I image the lung? 10 minutes, 2 hours, or how many cell type can you image at the same time? Okay, so we can image the lung up to 2 hours in one imaging session with one anesthesia. And then if it's necessary, we can repeat this lung imaging in the same mouse model because we can, suture up the mouse model after the imaging and then, recover it from the anesthesia. And then the next day, we can image that mouse again. And then, but although it's not easy, but we can do it. We can repeat this long imaging in the same mouse model. And then we can image 4 cell types because we have a 4 color detector. So we can simultaneously detect 4 fluorescent colors. So we can see 4 different cell types at the same time. And then another question was for breast culture imaging, where is the CD31 signal coming from? Do you use antibody labeling? Yes, we use we inject intravenously inject anti-CD31 antibody conjugate with the Prof. So as I explained in previous slides, so we inject this anti-CD31 antibody conjugate with the Prof. In this case, it's Alexa Fluor 647. So and then for everyday imaging, like this one, for everyday imaging of CD31, we inject the antibody every day. And then another question was about the pancreas imaging window. So yeah, although I skipped, so our pancreas imaging window is look like this. So if you look at here, so it has a ring, it looks like a ring, but it has some base in the middle. So the spleen, so if you look at here, this part is spleen and this part is the pancreas. But in the mouse, the pancreas is look like a sheet. So what it did was we put the spleen into this space, okay, like this, and then we just stretch the pancreas over this space, and then we inject our panco2 GFP cancer cell around here, so we can grow into this space. Okay, this is how our pancreas imaging window look like. Okay, and then another question is, is there any limitation of this in vivo live imaging compared to fixed tissue-based imaging? Yeah, of course, there are many limitations. The obvious limitation is labeling. So in fixed tissue-based imaging, you can label almost everything, right? But in vivo condition, either we need reporter mouse, like a GFP or RFP expressing reporter mouse, or we can use antibody. to label the specific cell types. But I only showed you several. I showed you several antibody example like Intravital, anti-lysis antibody or anti-CD31 antibody. It works very nicely, but not every antibody works in vivo condition. So that's obvious limitation when we do this in vivo imaging. Okay, and then the next question is, so it's a long question, but at the end, can we image the hippocampus after exposing the brain? After exposing the brain? Yes, we can actually. So there is already one very nicely developed technique. It looks like this. So what people can do, and what we can do, which we can aspirate the brain cortex and then insert this cannula-shaped imaging window. Okay, so we can aspirate the cortex and then leave the hippocampus intact. Then we insert this cannula ring into the brain, into the mouse head and into the brain like this. And after this setting, we can image the hippocampus. And then, well, it has been demonstrated by many other groups as well. So the simple answer is yes. And then the next question is, if we do everyday imaging for mouse, do we need to do surgery every day? Not necessarily. For example, in this case of imaging window cases, we only need to do surgery one time at the first day, at day 0. After implanting this imaging window, we don't need any additional surgery. We can just need to anesthetize mouse every day, to obtain this kind of images. Okay. And then the next question is, how do we go back to the same place for imaging? Yeah, like this. So although I only show these very high resolution images, we always start with the low magnification objective lens. So we see much wider area. And then we normally try to see the large vessel. Okay, so we can always see we normally in every cases we try to identify the large vessel by using lower magnification objective lens like 2x, like 2x or 4x. And then once we find those kind of large vessel, then we can use those large vessel as our landmark to find the exact same location in the same mouse model. So after we use larger and the lower magnification objective lens, we gradually change our objective lens to higher magnification, and then just look around and then find the same locations like this. Okay, and then the next question is, how do we deal with the motion artifact from breathing during pancreatic imaging? Do we fix the pancreas to the peritoneum to assure that you image the same region during every sessions? Yeah, I think I already answered actually. So for the pancreas imaging, we use this pancreas imaging window. Okay, and then this imaging window itself is fixed with this metal frame like this. So window is fixed, and then pancreas is fixed inside this imaging window on this base plate in the middle of this imaging window. So we don't have motion artifact due to breathing, and this is for the pancreas imaging window cases. But in other cases, we have other options actually. So in case of, for example, this is kidney. So this is the motion artifact due to breathing. Okay. So in case of this kind of motion artifact, what we do was we just record the video, the real-time video, and do the image registration, frame by frame image registration to compensate the motions. Actually, this function is already integrated in our microscope. So, and then this image registration is accelerated by using GPU processing. So you can do it in almost in real time. So this is one example. So this is moving tissue, and then this is our real time video. And then we do was we just activate our motion compensation function and just capture button. And then by using GPU processing, we can do the image registration in 3 or 4 seconds like this. And then you can generate this high SNR motion registered image registered images, motion compensated images. Okay, and then what's the maximum field of view of this live I-beam tag? What's the imaging resolution? Okay, well, the maximum field of view, to increase the field of view, we just use low magnification objective length. But in that case, so if we use low magnification objective like 4x, then our imaging field of view will be like 2 millimeter. But in that case, the number of pixels will ultimately limit our imaging resolution. So in our microscope, we can increase the pixel number to 2048. So in that case, so if we use lower magnification objective length, then the pixel size will ultimately limit the imaging resolution. Okay, I hope this may answer your questions.
Thank you very much for your kind answers to the questions, Dr. Kim. So finally,
and then for Reza's questions, maybe we should set up another meeting.
yeah,
one-to-one
sure.
meeting.
Sorry about it. Okay, so for the closings, I'd like to share the messages that our journey today has been both informative and enjoyable, thanks to your active engagement. And before we wrap up, a special announcement of the Intravital Imaging Workshop in Brigham Women's Hospital in collaboration with Professor Abdi's laboratory. So it is planned for the March 11th to 15th at Brigham Women's Hospital at 221 Longwood Avenue, Boston. So anyone who wants to experience intravital imaging technology will be welcome to perform in vivo imaging of your interest target on site. So please register through the pop-up link after the webinar finishes. And if you are still curious or have additional inquiries, don't hesitate to reach out to us at the email address information@ivimtech.com. So thank you everyone for being part of this enriching experience. Thank you very much.
Okay, thank you. Thank you very much.
Thank you.
Yeah. Okay, later. Yeah, let's have another meeting later time.
Yes, yes.
Okay.
I'm emailing you
Okay.
now.
Great.
Thank you.
Thank you. Thank you, everyone.
Bye. Bye-bye.
Bye-bye.